from the original Mexican center of origin, and those tend to be zucchini types. This is a spaghetti squash that's homozygous for the B gene. This is the North American cucurbita pepo, tends to be patty pans, delicatas, and acorns. These also have the B genes in them. These are representatives of cucurbita moschata, which is believed to be a northern South America origin. More of a tropical squash, not quite as well adapted to the temperate climates. These are probably the cucurbita pepo is the economically most important squash, followed by moschata, and then this is cucurbita maxima over here. But we've got a Japanese type, kogiku, some varieties that are probably have a butternut background, although I'm not sure, that have various sources of the B gene in them. This is a butternut type. And then here we have cucurbita maxima, which is a sort of South American origin in the more temperate areas in South America. Of course, the kabocha type, or sunshine, which has the B gene as well. The experimental Johnny's line, and then this I've been growing for a couple years now is Zapallo del Tronco, which is the only that I know of, the only cucurbita maxima that's grown for a summer squash, and it's really popular in Argentina. It's kind of semi-vine. The fruits are harvested much smaller than this, and I've been kind of interested in this, and I've been doing making some crosses with it. And then probably the least economically important species is cucurbita argyrosperma, and I have like green striped cushaw and Tennessee sweet potato and some varieties I found on eBay called yellow that I have no idea about. But I'm interested in different sources of this gene that causes depletion in chlorophyll. And so to back up a little bit farther, the pepo B gene, the B gene that originated in cucurbita pepo, came from an ornamental gourd. Not like this one, this is an egg gourd, but a spoon gourd. And a researcher named Ovid Schifris became fascinated with that gourd in the 50s, and he stabilized it for this trait that causes fruit to be yellow. The original lines that he had segregated all over the place. He couldn't figure out what was really going on, and he eventually created varieties that were fairly stable for either yellow or bicolor. And then he took those ornamental gourds, and he crossed them into modern cultivars of cucurbita pepo. So zucchini types, patty pans, acorns. So this is the first zucchini that ever had the pepo B gene crossed into it. It's Burpee's golden zucchini, and this is homozygous for the B gene. And nowadays you see tons of yellow zucchini, and they all are homozygous. Some of them are heterozygous or F1s. And sometimes the only way you can tell the difference between a homozygote and a heterozygote is just by the color of the peduncle. The peduncle will just be green on an F1, I've noticed. This is a kind of a famous variety called Orangetti from Harry Paris out of Israel. And this variety is also homozygous for the pepo B gene, but it also has another gene called L2, which originally comes from Ford Hook zucchini, just a dark green type zucchini. And that combination of L2 and the pepo B gene increased carotenoid levels 15-fold in this variety from the spaghetti parent. So another quick thing is this is the first acorn squash to ever have the B gene introgressed into it, and this is Jersey Golden acorn, which is not super stable. Okay, so that's a little background. So basically what we wanted to do is we wanted to take every source, there's two main sources of the B gene that have been studied extensively. The pepo B gene and the Maxima B gene. The Maxima B gene has been pre-existing in Cucurbita maxima since who knows when. It's believed to be, a separate mutation. So the early studies that Ovid Schiffers did is he took this gene and he took this gene and he crossed them together. But because you have such strong interspecific barriers or fertility barriers between Pepo and Maxima, he needed what they call a bridge species to be able to do inheritance tests. And so what they did is they did interspecific crosses between Pepo and Moschata and between Maxima and Moschata, and then they basically back-crossed to the recurrent parent for many generations until they had a stable Moschata line that was homozygous for this gene and that was homozygous for this gene, but was basically the same with all the other traits. And so he took those two parents and then he crossed them together. And the reason he did that is he wanted to find out if they were two separate genes or if they were alleles of the same gene. It's called an allelism test. So basically, I wanted to do the same thing because, 15 years ago, a researcher in Puerto Rico, Linda Wesselbeaver, sent Jim a new source of the B gene that had never been studied, that we believe comes from Cucurbita moschata, from a Colombian landrace. Jim received that material and he crossed it into Tromboncino lines, which is where that Y1 came from. And so we had this new form of the B gene and a new source, but we don't know the relationship of this B gene, the Moschata B gene, and the Pepo B gene, or the Maxima B gene. And so what I'm doing is a new allelism test where I crossed a homozygous parent that Jim had with not the B gene in Cucurbita Pepo that came from Pepo, but the B gene from Cucurbita Pepo that was now in Moschata, so that it was compatible. So basically, I took these two parents, the one that originated from Moschata and the one that originated from Pepo, but in the Cucurbita moschata species, crossed them together, got the F1, self-pollinated that in the greenhouse, or in the field in our case. And then this year I'm finally growing out the F2, and I'm growing out these huge populations of between 200 and 300 plants. And based on the segregation ratio, I can determine whether or not they're alleles of the same gene or two separate genes. And so that's what I've been spending the last 3 months doing, tracing squash vines, sometimes 20 or 30 feet. And I not only phenotype the fruit, but I'm phenotyping all the other aerial organs, the stem, the leaves, because these traits affect more than just the fruit. I not only made an allelism test between the Pepo B gene and the Moschata B gene, but I also did it between the Maxima B gene and the Moschata B gene. And here's an example of the Maxima B gene in a Moschata. And so, more or less, what I did is I took these two parents and crossed them together and did the exact same thing. And that's what you're looking at here, actually, is the F2 progeny of a cross between these two. And basically, what we found is that first of all, what we needed to know is if we had green segregants in our F2 generation, that would prove, if we had the right ratio, we needed 3 out of every 16 plants to be a green segregant. If we had those green segregants, that meant they're two separate genes. If we didn't, and they were all precociously yellow in some way or another, that would mean they were genes. That would mean they were alleles of the same gene. And so what we found is we did have roughly 3 out of every 16 plants in the Moschata B gene by Maxima B gene cross, but not in the Pepo B gene by Pepo B gene by Moschata B gene cross. And so what that tells us is, we think, I haven't really analyzed the data yet, but I've looked at it. What that basically tells us is the Moschata B gene and the Pepo B gene are alleles of the same gene, but the Moschata B gene and the Maxima B gene are two separate genes. And And so that, that will help with future breeding. And I think you might ask, why do this? I think really the main reason is kind of what you guys were noticing, is that first of all, with summer squash, people love the color. It's really appealing. And then with winter squash, if it can increase carotenoids 15-fold, you can really create a more nutritious squash. There's been some data that shows that it improves flavor and other benefits. But I'm to the point where I'm just like a mad scientist. So I've been making crosses. This is the last one. So I believe that I found a new form of the bee gene that hasn't been studied. It originated in our gyrosperma, and so what I did is I repeated the same process where I crossed this with Moschata, Maxima, and Pepo bee gene, because our gyrospermas are compatible with the Moschata species, so you can make crosses. That's what I wanted to show you. So if you guys want to look at these real quick, these are some examples of segregants. You guys can pass them around. That's a segregant between the Moschata bee gene and the Maxima bee gene. And so these are the type of things I'm looking at in the F2 generation. So you'll see traits on the stems that have incredible depletion of chlorophyll. You can see that. So there are different expressions between the three different alleles in bee genes. The Maxima tends to always have this chlorophyll depletion in the stem, the petioles, and the leaves. It's really, really pronounced. You can see what I believe is a homozygous fruit. So in this case, I would know it's homozygous for the Maxima bee gene, but I would not know whether it had the Moschata bee gene at all, necessarily, because it might not be expressed. That was another thing I failed to mention. The Pepo bee gene is a dominant gene, the Maxima bee gene is a dominant gene, and the Moschata bee gene is recessive. So that really changes the way the segregation ratios or the traits are expressed in the heterozygous state, as you can imagine. But yeah, here's just some examples of the traits. They're kind of wilted now, but of the Maxima bee gene in the Moschata background. Segregants that, so it's hard to explain, but if you see the similarities between these two expressions, you can see sort of a solid line. This came out of the F2 segregating population in the Maxima, and this is the parent. And you can see how similar those are. So when I'm phenotyping, I would say, this is probably doesn't have the Maxima bee gene at all, and it's homozygous for the Moschata bee gene. So that would be my hypothesis as I go through every single plant in the field, flag by flag. Every flag represents one plant, and they're all numbered. And so I had to do that for about 5,000 plants. So hopefully when I'm done with this, I won't hate squash. But at the moment, I'm still loving it. Here's another example of a Maxima bee gene by wild type cross. Yeah, it's really been incredible. I'm really enamored with this trait. I hope that we find more ways of increasing quality.